Okay, well, good afternoon. Sorry for such a abrupt introduction. So we will continue where we left off yesterday. And so yesterday we concentrated mostly on defining the question that we may want to answer with targeted proteomics, with quantitative targeted proteomics experiments. And we talked about the experimental design. And so we talked about the fact that there are three fundamental principles of experimental design. It's replication, randomization, and blocking. And so these three fundamental principles need to be considered before we conduct the experiment, before we even plan out the entire study. And so the goals of replication, randomization, and blocking is to avoid a bias, so a situation where our conclusions from the data are systematically incorrect, and also maximize the efficiency or minimize the inefficiency of the experiment, which is translated into the variance of the differences that we look into, right? So that was our main topic last time, and so we started talking about some basic methods for detecting differentially abundant proteins, and so I would like to review this once again. So this part that we started on just talking about basic t-tests and assumptions and so on, and then we will talk about a couple of additional topics today, which is what happens when we work with more than one protein at a time, which is what we normally do, right? And also I will start introducing relative quantification of proteins with the tool that we develop in my lab, which is MStats, and I know that later this afternoon we will have a more detailed tutorial and also a practical hands-on exercise on MStats as well, so I will just start the introduction. Okay, so let's revisit once again what happens when we are interested in detecting differentially abundant proteins. So proteins that change on average between healthy and disease groups, on average over multiple subjects. And for the time being, we just think about a very simple situation where we have proteins digested, maybe label-free, LCMS experiment where we have one peak per protein per patient, just to be very simple, right? And so our question is whether the mean of the log abundance of the feature in the healthy group is systematically different from the mean of the log abundance in the disease group, so more systematically than as would be expected by random chance. And so the way we kind of set this up is to say, well, our question is really not regarding these particular means which we observe from the data, but regarding the population parameters. So this will be the means of every single subject who has the disease in the underlying population, and the mean of every single subject in the control population. Of course, we don't know what these means are because we cannot measure, we cannot quantify the protein in every single member of the population, but we can make some guesses. And that's really how statistical inference works. We will make a guess, say, well, let's assume that in the underlying population there is no change in protein abundance between the groups. And so if we assume that, we can characterize the properties of the data, which would be expected if it was the case, and then we will see if the data actually corroborates that or not. And if there is an evidence against that, then it indicates that our assumption was wrong, and therefore there is evidence for the alternative assumption, which is there is the difference between the means in the populations. Okay, and so that's how it goes. So we start by assuming that there is no difference between the groups, and we will see if the data supports that. And the quantity that we derive from the data is essentially a signal-to-noise ratio, right? But in this case, the signal is the biological signal, and the noise combines the biological and the technical variation. So it's not just the measurement, right? It's the entire biological variation which contributes to the noise when we talk about the difference between the groups. And so the signal will be, in this case, the mean of the healthy group minus the mean of the disease group. And then the noise is the estimate of variation combined from both groups, right? So here we have the estimate of variability, which is just the sample variance, right? Which is very simple. And so now, so S1 will be the variance associated with the disease group, S2 would be the variance associated with the disease group, N1 is the number of replicates in this group, N2 is the number of replicates in this group, okay? So this is just the variance, but the ratio of the variance to the sample size is the variance of the mean. So this characterizes how confident we are in the individual mean of the group. And so because the sample size is in the denominator, it means that the larger the sample size, the more confident we are in the mean, right? So the smaller this variance becomes. And so as the sample size grows, we are more and more confident in the mean of this population, or in the mean of this sample, right, which we actually observe. And so then if we want to look at the variability associated with the difference between the mean of the healthy group and the mean of the disease group, we combine the uncertainty in the first mean with the uncertainty in the other mean, and then we just take the square root of this to take it back on the original scale as opposed to squared scale. And so, well, that's really what's going on. And so just in terms of the terminology, right, so this will be the variance of this group. S1 without the square will be the standard deviation associated with this group. But the square root of this quantity is the standard error of the mean, right? So there's a difference between the standard deviation and standard error. Standard deviation characterizes the spread in the population. Standard error characterizes how confident we are in the mean of multiple replicates. And so as we have more and more replicates, the standard deviation will kind of stabilize, but the standard error will go down, right? So that's what we talked about. But kind of, if this sounds confusing, so fundamentally this is the biological signal divided by the biological noise. So that's what we're talking about. So another thing we discussed last time was that the assumption that we will make is that the mean of this group and the mean of this group, if we were to repeat this experiment many times, overall it would be roughly following the normal distribution. So it will have this kind of bell-shaped histogram. And this assumption is plausible, and so this was the picture from the series that I appreciate so much. So this is the points of significance collection from Nature Methods, which has 2-page vignettes on different topics. And so this was actually the article which was called The Importance of Being Uncertain. So what is the importance of having different extent of variability in the data? And so if we start with the data which follow normal distribution, so if we take 3 data points from this distribution and calculate the mean and do it many, many times, so the mean, well, clearly will be more narrow than the initial data, right? And then as sample size increases, it becomes more and more concentrated around the true mean in the population. But the comment is often made in proteomics, well, actually, how do I know that my data are normally distributed? Maybe I have some outliers, maybe I have some kind of unusual things, and so on. Turns out that it doesn't matter if the data are normal or not, so the data can come from some kind of really bizarre shape. But if the sample size is increasing, so the histogram of the means actually becomes more and more bell-shape formed. And so this is the assumption. We do not necessarily assume that the data are normally distributed, we assume that the means are normally distributed. Now, clearly, the larger the sample size, the more plausible this assumption that the means are normally distributed. So this is one of the reasons why it is good to have a large enough sample size so that our assumptions actually have some ground. But even if we start with something really bizarre, eventually the means themselves actually are roughly bell-shaped. So this is a good thing. And the name for this is central limit theorem. So as the sample size increases, the mean of many data points becomes kind of following more and more of a bell-shaped distribution. Okay, so that's one assumption that we make, right? So now what we said is, okay, we calculate the signal-to-noise ratio for the means. Now we also assume that the individual means follow normal distribution. And so the sample size helps us kind of to make sure that this assumption is plausible. Now, here's another role of sample size. So oftentimes in proteomics, we have studies with 3, 4 replicates and so on. And so in addition to kind of being more in this situation, we are less sure in the mean, we are also, sometimes we tend to underestimate the variability in the underlying populations. And so this is from the same vignette. So here we have the x-axis is the sample size. And so here is the sample mean, which is calculated by taking that many replicates from this distribution. And so when the replicates is small, you see that the mean is kind of quite variable, right? So it goes all over the place. But then when the sample size increases, the mean becomes very close to the underlying mean in the population. But this is what happens with the standard deviation, that as the sample size is small, standard deviations tend to be smaller, which means that we are actually too optimistic. We underestimate the amount of variability in the data. And if we underestimate the variability, it means that we will be too optimistic. So we'll often mistake some signals for systematic signals. So we will end up making more false positive decisions than there should be. And the reason is that if you only take 3 data points, so chances are that you will take them from somewhere in the middle of this histogram, and so you would not be seeing the lower tail, you would not be seeing the higher tail, and you would think, okay, well, this is the extent of variability I have in the data. And only when the sample size increases, you actually realize that you had also lower values, and you also had higher values, and eventually standard deviation will converge to the true standard deviation in the population. But so the danger of small sample size is not just the fact that the assumptions do not hold and things like that. I think this is actually much more important, that it just simply leads to more false positives because we underestimate the variation in the underlying population. And this is really the most important part. You just don't have enough data to characterize how much noise you have in the entire system. So the more data you have, the more you realize how much noise is actually there. And so this is again a standard error, and so you see that standard deviation converges again to some constant, but standard error actually goes to 0. And so when we talk about the estimates of variation, it is really extremely important to distinguish standard deviation from standard error because you expect different patterns from this, right? And so it kind of very similar wording, right? But they're very different concepts, so we really have to be very careful with that. Okay, so that's what's going on. So now, well, we have the signal to noise, right? So we now assume that the means are normally distributed. We kind of realize that large sample size will allow us to characterize the noise in the best possible way. But now we need to make the decision. So how large is large enough? So again, we have this difference between the groups. We have the estimate of variation. So how large signal to noise should it be to actually claim that this difference is more systematic than as expected by random chance? And so the reason why we make all these assumptions, that we assume normal distribution and then we assume that the variability is characterized correctly, is that if all these assumptions hold, and if we assume that there is no difference between the groups, without seeing any data, analytically we can predict what is the plausible range of values of signal to noise, which is consistent with this situation. And this is really why it is done, because then we know that if there is no difference between the groups, if the normal distribution is plausible, and if the variability is characterized correctly, then the plausible range of values of the signal to noise quantity will have this particular shape with some very specific spread. And so then what we can do is to say, well, we can kind of use this range of values as a benchmark. So now if we actually calculate this from the data and it falls somewhere in the middle here, we say, yeah, well, that's what we expect analytically, right? Okay, now theoretically. This means that the data has no evidence against what we assume. So there's no evidence against the assumption that there is no difference between the groups. On the other hand, if you calculate this and it falls somewhere here or somewhere here, well, you say this is very unlikely that it would be kind of coming from this particular distribution. It's too extreme. Then we say, well, then this distribution is not plausible. So this is evidence against what we assume. So this is evidence in favor of the alternative hypothesis that there is a difference between the groups. And that's exactly how it goes, right? And so the last piece that this is missing, we say, well, so how extreme should be to call it extreme? And so usually we select a quantity which is called false positive rate, and usually it's 5% or 1% or something like that. And so the area under this distribution is the probability of being in this range. And so we will have cutoffs such as this will be, let's say, 5% divided by 2. This is also 5% divided by 2. So together this area plus this area is 5%. And so now we say, well, if the quantity here falls in this tail or in this tail, we say that it's evidence that it's too extreme, so it's evidence against this assumption that the groups are the same. However, if it falls within this range, then we say, well, there's no evidence, it's kind of as the type of value that is expected. And so that's it. So we select this cutoff, which we have to do before collecting the data, and it's usually very kind of literature specific, but 5% or 1% will be quite typical. And so if the signal to noise falls in these tails, then we reject the null hypothesis. So that's really how we make these decisions. Now there's another equivalent way of doing this, is instead of selecting the cutoffs, we can actually overlay this observed quantity on this graph. And so that's what it looks like. So we usually overlay the value, and then because it's symmetric, we overlay them minus this value as well. And so now, well, if these observed values are in the tail, we can calculate the area to the right of the observed value plus the area to the left of the observed value. And this is the p-value of the test. So the p-value is the probability of seeing something more extreme than we actually observe from the data, if we assume that there is no difference between the groups. And so this is on the graph, it is the area to the left of this blue line plus the area to the right of the blue line. And so clearly, right, if the area, so if the p-value is less than 0.05, then we reject the null hypothesis. It's evidence against the null hypothesis. But if we are here, then the area to the left plus the area to the right will be greater than 0.05, then there is no evidence against the null hypothesis. So these are two equivalent ways of doing the same thing. So either just looking at the signal to noise or looking at the p-value of the test. And so this allows us to make the decision. So so the good thing is that it takes into account both the difference between the groups. Remember, this is the log-fold change, right, in our experiments, and the estimate of variation. So this is better than just using fold change cutoff, right, because it accounts for how much variation you have on the data at the same time. So that's what's going on. However, remember we talked about small sample size and the fact that if small sample size, we underestimate the variation. Well, so these problems propagate themselves all the way through the tests. And so here's another example, so from a different paper, but also from Nature Methods, which talked about that when we have small sample size, estimate of variation is not reliable, and therefore p-values themselves are not reliable. And so here's an example, so where all the assumptions verify, so they have a very small difference between one group and another group, but it's a real difference. And now you have, well, reasonable variation around the mean in each group. And so what they do, they just repeatedly draw data points, I think it's 10 per group, from these two populations. So this will be experiment 1, this will be the independent replicate of this experiment, this will be again another, so 4 independent replicates of the experiment. And because the overlap is very large, and because the sample size is fairly small, just 10 and 10, so you see that in this first experiment you have p-value 0.005, but then just by random chance from exactly the same population you have 0.8, a completely different conclusion. And here it's again not different, and here it's again somewhat different, right? So because the sample size is relatively small as compared to the difference in means and then comparing to the underlying variation, so clearly this is what is called the experiment is underpowered, because the variability compared to the difference in means is too large. And the sample size is too small to characterize that. And so if it's underpowered, it means that your results can change dramatically from one experiment to another. And so this is another reason why sample size is actually very helpful. So if you have a small-scale screening experiment with 3 replicates in one group and 3 replicates in another group, well, we may often have situations where the changes are fairly subtle. So don't be surprised that if you redo it again, you have a different conclusion. This is just kind of the consequence of the fact that we have large variation and small difference in means and a small sample size. Okay, so I guess my point is take it with a grain of salt, right? So if you see 3 replicates in one group, 3 in another group, don't kind of expect every protein with small p-value to be reproduced exactly as is in the next experiment. So there's quite a lot of uncertainty just coming from the overall situation. Any comments? Right? Yeah, I have a question.
So, Olga, the number of replicates that you show here to stabilize this type of tests are normally above 10, right? And except for biomarker discovery or validation experiments, so most of the basic research that is done with proteomics has 3 or 5 replicates per conditions. So are all our experiments underpowered?
Well, the answer is that it's an interplay between how big is the change, how much is the variability around the change, and how big is the sample size. So if you have an experiment when you have a 20-fold change, you will probably see it with a naked eye, right? Even like with 1 or 2 replicates, and then you would repeat it again and you will see it again, right? On the other hand, if you 1.1-fold change, most likely it will be underpowered, right? And so most likely you'll be in this situation where, depending on how lucky you are, it can go either way, right? So it's really a combination of the variation, the actual effect, and the sample size. There are really in dependence between those three things. Of course, the problem is that we have one sample size for many proteins, and different proteins have different changes, right, and different variations. So most likely for many proteins, most experiments are underpowered. And so when you're doing an experiment with sample size 3, you're essentially speculating that at least some proteins will have a really large fold change that you will see no matter what, and it will be reproducible. So you are really betting on the fact that in your whatever treatment or stress that you have, you will see some really substantial fold changes which you will see and which will be reproduced. How often it's actually the case? Well, I don't know. And I think sometimes we're maybe betting too much on that. But you know, then again, it's kind of a trade-off, right, between the costs and all of that.
Is there a way that you can compute the fold change that you can detect with a certain number of
Yes.
replicates?
That will be towards the end, yes. And if we don't get to this, we will talk about how to do it in MS stats, yes. But the thing is, and that's actually an interesting point, so you cannot really, so sample size calculation is part of the experimental design, right? And so this is something you do obviously before collecting the data. The problem is that you need to know a lot about your experiment to do this. So we'll have to kind of work through one example data set, and then I will kind of take it all the way back to the design and how we can talk about sample size calculation. But I hope that that's actually relatively intuitive, right? That it's really an interplay between multiple things, which means that if somebody comes to me and says, well, I'm starting a new experiment, is 3 replicates enough? You know, I don't know, right? It depends on how much variation you have, it depends on how big fold change is, right? So you need to know a lot.
Is it correct to set up for, let's say, fold change threshold, and in combination with the p-value, and that's how we select our
Correct.
proteins?
Correct. And so what, well, maybe it's a good time to mention this. So you would use the p-value cutoff to make a decision if this is systematically changing or not. And this will be statistical significance. But on top of that, well, nothing prevents you from also making a fold change cutoff, right? Maybe for the purposes of your experiment, you're not interested in fold changes below 2 or below something, right? And this is what we call a practical significance cutoff. The important thing is that you apply statistical significance cutoff first. And once you narrow down to the list of statistically significant things, then you can filter in any way you want, including by the fold change. But you could not only use fold change. That would not be right.
Thank you.
Okay, so now we had a question last time, and I'm really happy to have a chance to talk about this. So in addition to making the decisions in terms of significant or not significant, sometimes you want to just characterize how much variation you have in the system without just kind of declaring black and white, right? Significant or not significant. And so a good way to do this is with error bars. And so the important thing here is that there are at least 3 types of error bars that you can draw, and they convey very different type of information. And a common mistake that I see that people will just have a paper with an error bar in it, and you have no idea what it means, and therefore you have no idea how to interpret that, right? So again, the point here is when you do plot error bars, make sure you specify what this actually is. And so one very common approach, which I think is probably among the best approaches, is to plot confidence intervals. So this is again from the same series of point of significance. So the definition of a confidence interval is that it's an interval around the estimated log fold change, such that if you repeat your experiment 100 times and use the same procedure to derive this interval, then in this case, 95 out of 100 intervals will contain the true population mean. And 5% will be outside, so they will not contain the true population mean. Now, how do we know that on average 95 and not 93 or not 92? Kind of for the same considerations here, if all the assumptions hold, such as normal distribution and correct characterization of variance, then we can actually analytically calculate what would be the percentage of times that this interval will not contain the true mean. So again, it is based on the analytical calculations and the assumptions that we have. But so the definition of the confidence interval is that, for example, a 95% confidence interval will cover the true mean 95% of times. If we were to repeat the experiment, collect the data in U, use exactly the same procedure to calculate the interval, 95 times out of 100, this interval will contain the true mean just from the analytical calculation. And so here's the kind of comparison between the 95% confidence interval and the standard error of the mean. Okay, so the dark lines here, this will be the confidence interval, and the light gray bars in the middle, these are standard errors for the mean. So clearly standard errors are much smaller than the confidence intervals, right? But the standard error only tells you about, well, what is your confidence in the mean? Whereas confidence intervals tell you how likely they are to contain true mean in the population. So these are very different interpretations. And so it is very tempting to plot standard errors of the mean because they look narrower. But if your question is, does my confidence, so if this is the true value is the difference between the means of two populations, and if you want to know if it contains 0 or not, you really need to look at the confidence interval and not at the standard error of the mean. And so this is kind of the point, right? So just to kind of repeat it once again, so if this is the true difference between the means of two groups in the population, and if we're plotting the confidence interval for the difference between the two means based on our data, so 95 times out of 100, these intervals will contain the true value. So if the confidence interval contains 0, then we can say we're 95% confident that there is no difference between the groups, right? And standard error of the mean or combination of standard errors of the means for the two groups will not be able to give us this conclusion. So if we want to make conclusions along the lines of, well, this is how much evidence we have that there is no difference between the groups, we actually have to use confidence intervals because this is what gives us the probabilistic guarantee for that. Standard errors of the mean do not give us the probabilistic guarantee for that. But the error bars look narrow, so that's why sometimes people like to show that. And this is the illustration. So, and the third type of error bar will be standard deviation, right? Which is just characterizing the spread in the population as opposed to the means, right? So that's yet another thing. And so again, this series, they have very nice plots. So what they do, they use sample size 10 all the time. And so they just kind of decided that, okay, let's look at the difference between the two groups. Let's say 1, right? So this is 0 and this is 1. And they manipulated the standard deviation in the population such that you produce exactly the same error bars if you plot standard deviations, standard errors, and confidence intervals. So this will be one type of experimental situation, this will be another type of experimental situation, and this will be another one. And they just kind of manipulated the standard deviations in the population to show that you have exactly the same width of these three types of error bars. Look at the p-values. If you plot confidence intervals which do not overlap, p-value is this. But if these are the standard errors of the mean which do not overlap, the p-value is 0.17. And if these are standard deviations, it's even smaller. So the conclusion in terms of whether the groups are different or not, for exactly the same looking error bars is very different depending on what you plot. So if this looks confusing, so I very much invite you to read this 2-page vignette which uses this figure and kind of has substantial text describing that. But for the moment, the point being is that if you have error bars which look kind of, they just barely touch and they're the same width, it can imply very different things in terms of are the groups the same or different, depending on whether these error bars mean standard deviation, standard error, or confidence interval. It will be very different conclusions. So make sure that you can explain what you are showing, right? And so if you have, for example, standard error of the mean that looks like that, don't get too excited because it doesn't mean that there is really a difference between the groups. Okay? And so this is another way to look at it. So now they actually have the same standard deviations in the three cases. And so now they calibrated this in a way that the p-value is the same. And so you see how different the actual error bars are in order to produce the same p-value, right? So if you look at the confidence interval, there is partial overlap. Standard errors of the mean have to be very, very different to lead to the p-value of 0.05. However, the spread of the data points in the population can have quite some overlap because we're comparing the means and not the individual observations. Okay? So I realize that it may be somewhat kind of obscure, but the point is it matters. And in these vignettes, you can read more about that to kind of clarify it for yourself in more detail. Okay? And so here's just another situation. So again, they just show for different p-values what would standard errors of the mean look like versus what the confidence intervals would look like. And so generally, you can tolerate a lot more overlap in confidence intervals than you can tolerate overlap in standard errors of the means in order to have this type of p-values. Okay? Any questions?
Is it working? Yeah.
Mhm.
So would you say that, I mean, it seems from this, the least useful one is the standard error in terms of interpreting just from a visual. So is there a proper use case for this, or would you sort of caution people against using this?
Well, it depends on the point you're making, right? So if you want to talk about, well, this is how variable my population is, standard deviation is appropriate, right? If you want to say, well, this is how confident I am in the individual mean, standard error is fine. But if you want to make specifically the claim that these two groups are different,
Yeah.
then you have to talk about confidence intervals. And it's true that in most cases we would probably want to emphasize the difference between the groups when we're doing it, right?
Right, so for
In which case it's confidence interval.
So for the people in this room, the standard error is probably not particularly useful for the majority of comparisons. I mean,
it's
the
fine
most
to
common
show
use
it.
cases.
Right, but the problem is that people will jump right away to saying, oh, look, this is very different, right? So they kind of tend to interpret the bars as some kind of evidence for difference between the groups.
Yeah.
And if that's the case, then you really need to speak in terms of difference between the groups, which is confidence interval.
Okay.
Okay, so this is one important thing. So now the other important point that I want to make. So t-test is just a very standard statistical technique which has been around for, I don't know, a very long time. So now we're moving closer and closer to a situation which we actually have to deal with in targeted proteomics. And one aspect of targeted proteomic experiments is that we don't just work with one protein, we work with many proteins at a time. And well, it depends on the experiment, right? If we do SRM, maybe we have several dozens. If we do DIA, we have a couple thousand, right? But essentially, always we have more than one protein in the data set. So what happens in this case? So how does it affect our ability to make decisions? So let's consider a situation where we have two proteins, just very simple. And I do two because I can draw it, right? So let's say this is protein 1 and this is protein 2. So now for each protein, I have the assumption that there's no difference between the groups, and I'm looking for evidence against this assumption. And so for protein 1, I would have this, I just flipped this distribution. So this is the theoretical range, right? The theoretical distribution of values that I expect if in reality there is no difference between the groups. And so I set up my alpha, right? So now this is my rejection region. And so regardless of the value of protein 2, so no matter what protein 2 is, I will make my decision for protein 1. And the combined area here plus here is 5%. So I have a 5% of chance of claiming that there is difference when in fact there is no one. Okay, fine, right? This is exactly what we did so far. But now I do exactly the same thing for the second protein. And so now for the second protein, same thing, right? So I have this plausible range of values. I set up my cutoff in the tail. So I have this area plus this area is 5%, regardless of the status of protein 1. And so I have a 5% of chance of claiming a false positive difference. Fair enough. The problem is that we're not doing one and the other separately. They're both part of the same experiment. So now if we're trying to make conclusion for both proteins at the same time, I actually have this situation where I have the decision to make for protein 2. I also have a decision to make for protein 1. But now the probability of making a mistake is actually the combined area for these two proteins. And so I just said that this is 0.5. I also said that this is 0.5, so the combined is clearly greater than 0.5. So now, if I have two proteins, my probability of making at least one mistake, right, of claiming incorrect false positive is larger than what I was trying to control when I was working with one protein. So you can see already that this is greater than 0.5. Now we don't have two proteins, we have maybe with DIA 2,000 proteins. So for DIA, and when we have large numbers of proteins like that, the probability of making at least one false positive is essentially 1. You're essentially guaranteed to make at least one false positive discovery just because when you look hard enough, you will end up finding something by random chance, right? So if you have 5% to make a false positive, so out of 1,000 tests, right? So how many? You can have 50 false positives right there, right? So, so the way to deal with that is to reduce. So the way to reduce the probability of making false positives is to reduce the rejection area, right? So it means pushing the decision boundary from here all the way back there such that the area outside of the dashed line now becomes 5% or 1% or whatever we would like to do. But this essentially means that now we have to become more conservative, because now it becomes more difficult to reject the null hypothesis for each of the proteins, because now we require stronger evidence, more in the tails for each of them, such that together we can control the probability of making a mistake jointly. And so that's what happens. And so there are multiple ways of doing this, and you may have heard about Bonferroni approach. So this is exactly what it's doing. So it's reducing the area outside of the dashed line, and we do this by dividing this alpha divided by 2 by the number of tests. So if we have 2 tests, so now I take alpha divided by 2, I divide it again by 2, so it becomes alpha by 4. So I have to be more in the tail. But if we are in the IA, we have 2,000 proteins. Now I take 0.05 divided by 2 divided by 2,000. So this would require some kind of incredibly extraordinary evidence to claim that even something is changing. So this is the case where the large number of proteins leads to a decrease in our ability to find any differences just because of this multiple testing issue. And so things like Bonferroni are not really appropriate for the type of experiments that we talk about here. So this is not practical. So there was a really big contribution 10 plus years ago in the statistical literature, where people said, well, you know what, here we are controlling the probability of making at least one mistake. So we want to have a very high probability of making no mistakes. That's too much to ask from very high throughput experiments that we have. In fact, we're maybe okay with having few mistakes, as long as we can control the proportion of mistakes that we make in the entire list of differentially abundant proteins. And so this is what happens. So the probability of making at least one mistake, this is what is controlled by the Bonferroni approach. And so the new alternative way of doing this is to control false discovery rate. And this is what it looks like. So this is the truth. So if we test m proteins total, and among these tests we have claim that our proteins are differentially abundant, and we claim that the remaining are not differentially abundant, well, there's some numbers here, right? So this is S is the true claims, right? So it's true changing and we claim as changing, and this will be the false claims, right? So these are true not changing, but we claim they're changing. The problem is that we don't know these numbers. All we know is how many we actually claim. But again, there's a statistical procedure proposed by 2 statisticians, Benjamin and Hogberg, which allow us to control the proportion of V in R. So we can control the average kind of proportion of false positives in the entire list of the proteins that we claim differentially abundant. And there is little technicality to avoid division by 0. In practice, we never have 0 claims. We have at least some claims. So essentially, we are controlling the proportion of false positives in the list of the rejected null hypothesis. And so Bonferroni expects V to be 0 with very, very high probability. False discovery rate allows V to not be 0, as long as we control the proportion of V in R. And because it's a less conservative criteria, it allows us to push this decision boundary not too far in the tail. So it allows us to actually see some differences. So it allows us to kind of be able to even consider moderate differences as evidence against the null hypothesis. So that's what's going on. And so, in fact, there are many other ways of doing this, but this is really the most kind of standard and well-recognized approach, so using Benjamin and Hogberg. And so what you can do, you can either, going back to my previous examples with p-values, you can either change your decision, or you can adjust the p-values. You can manipulate the p-values such that the p-value cutoff now considers the entire list and controls the false discovery rate. And in fact, this is exactly what MS-Stats is doing. So when you will have exercises, so MS-Stats accounts for that by adjusting p-values. And so this is exactly, to go back to this question, this is exactly what the outcome would be. So you have log-fold change on the x-axis. This is the practical significance. You have the minus log p-value adjusted for false discovery rate. So small p-value is good, right? And then it's difficult to see small values. So minus log of that means that high means significant. It's just to be able to see it better. So this is statistical significance. This is practical significance. And so at the very least, you want to have a statistical significance cutoff so that things above are statistically significant, so evidence against a null hypothesis, but then nothing prevents you from making an additional practical significance cutoff for fold change. So you're really interested in the left corner and in the right corner. That's what's going on here. Any comments on that? So this is also an important aspect of that. Okay, so now the last part for today is, well, we still need to move closer to what the data actually look like in targeted proteomics as opposed to what we can have in simple methods. And so we talked about the fact that we have many proteins, but there is more than that, right? Because my t-test example had 1 peak per protein per subject, which is not really the case. Plus, I only said, okay, let's imagine we have 2 groups, healthy and disease. In reality, we can have all kinds of complicated experiments, including time course, paired designs, and all kinds of things. So the reality is more complicated than just 1 peak per subject and 2 groups. And so the goal of MS Stats is to extend this type of techniques to a more general situation, which is actually common in targeted proteomics and in general in proteomics. And so here's an example. So you will hear more about this, so I will just do a very quick run through, and then in the afternoon you will have a more detailed discussion of that. So here's one example which actually comes from here from ETH, from the Ebersold lab, to illustrate complex experiments. So this was a study of breast cancer cell lines, and there are 2 cell lines, which is low and high invasive cell line. And the researchers were also interested in the response of the cell line to low oxygen conditions. So the cell lines were exposed to normoxia, meaning normal oxygen, and hypoxia, so this is low oxygen. And also, there was a different duration of exposure, so for 6 hours or for 24 hours. So you have 3 things, right? So invasive status, or cell line, oxygen exposure, and duration of oxygen exposure. So these are 3 different factors. And so for each of the factors, we also had different cell cultures and then different technical replicates. And so if you look at one protein, it looks kind of like that. So this, by the way, was the shotgun LCMS experiment. But again, for SRM, it will look something very similar if it's a label-free experiment. So here is the all combinations of all of these 3 conditions. And here each color corresponds to a peptide of this protein. And the dot will be the replicates of this peptide in each particular condition. So, well, what do we see? We see that the peptides generally kind of have a fairly similar story as far as comparing this group to this group. However, within kind of these parts, there's some disagreement. So this, for example, is a low intensity. Maybe it's a missed peak or misquantified peak somehow. Here it looks like there's some interference, right? And so the question we may want to ask is not just, well, is there any difference between any of the conditions? That's too general. We actually want to ask specifically, is the difference between normoxia and hypoxia for MCF7 the same as the difference between normoxia and hypoxia in this cell line? So do the cell lines have the same response to low oxygen, or do they somehow vary in response? Or is the progress in time under hypoxia for this cell line similar to the progress in time under normoxia? So there are all kinds of combinations of conditions which can be considered to answer various questions which are relevant to this problem. And so what was kind of done traditionally is that our collaborators would just pick out 2 of those conditions at a time, kind of look at them separately. And so what MS Stats does, it tries to extend the t-test that I talked about earlier to these more complex situations to leverage all the information which is relevant to this problem and do a better job, and also help answer more complicated questions. So that's kind of what we're doing. And this is the overall run through how MS Stats works. So, of course, the first important part is the experimental design, right? And before we do anything, right, we need to actually convince ourselves that we're interested in testing, as opposed to class discovery or class prediction. Because, as we said yesterday, these will be very different statistical methods. So here is specific to comparing the means of the populations. So meaning that if we have a small p-value from this method, it's not a biomarker, right? It's just the evidence that there is a difference on average. It doesn't mean that we can predict the status of the individual patient, right? We're still comparing the means of multiple subjects, but it can be useful, right? And so then, once we establish that, we also need to think about what are the sources of variability which will affect the intensities of the peaks. And it can be different types. So in this example, it's called a factorial design because we have 3 factors which are kind of intertwined, but we can also have a time course design, we can have paired design where the same person has a healthy, let's say, tissue specimen and a diseased tissue specimen, Time course means that you have profiles in time. So here's another example, which we had in collaboration with Indiana University School of Medicine, where this was a study of osteosarcoma. It's a childhood cancer. And so in this study, the researchers had plasma samples from controls at one time point, but then for disease subjects, they actually had samples before treatment, that's at some point during chemotherapy, at some point after surgery, and also at the very end, like when they were in remission, they also had some plasma samples. So the question was, well, multiple questions. So are the healthy subjects and the disease subjects at diagnosis, which proteins change? Then if these proteins change, do they temper down and kind of come back to the normal levels after the chemotherapy, or are they somehow affected by chemotherapy? At which time point they become changing? So there are all kinds of questions you can answer with this design, and so you cannot really do it with a pairwise t-test. You really need to model all the data jointly, and that's exactly what MS-STATS is doing. So, well, after that we go obviously into the whole experimental part, and once the data are collected, so there's a whole set of steps that we need to account for, and the first and very important step is quality control and normalization. And this is something that I cannot stress enough, you know, because it's so frequent that we just say, well, we have this automated pipeline, so we have the data going through, we have some p-values at the end, and that's what we do. It is extremely important to look at the data and see if something went wrong at any step of the experiment. And this can manifest itself by some kind of intensities in the runs which are completely off, or some kind of interferences, or standards which do not behave as you would like them to behave. And so MS-STATS provides tools to visualize things. So obviously Skyline visualizes this very well as well. And maybe I should point right away that MS-STATS is an open source R-based software which is interfaced with Skyline. And so we work very closely. Some of the methods are implemented in Skyline, but other methods are not implemented in Skyline. So it's kind of complementary to each other. But this will be one example of a quality control plot that you would use. So this would be now an SRM experiment. And let's say we have heavy labeled reference peptides spiked for each endogenous peptide of interest. And so the x-axis here is the run number. The y-axis is the log intensity of all the peaks in this run. And we look at references separately, and we look at endogenous separately. So here, each box will describe the spread of the reference intensities across all transitions and all peptides and all proteins in the run. And because these are reference standards, we don't expect any biological variation in them, right? Because we spike them in a constant concentration. So we expect them to kind of line up perfectly. And if they don't, it means that there are some kind of artifacts which occurred after the standards were spiked. And so a simple way to do this is to apply some kind of transformation to the standards to make them, to equalize their distributions, and then apply the same shifts to the corresponding endogenous peaks. And so now this will normalize. So this is a kind of situation where it was a targeted experiment, so many proteins are changing between the runs. So we equalized the references, and we applied the same shifts to the endogenous peaks to make sure that we eliminated some of the systematic artifacts which affect all the runs. And I know that there will be more discussion of this this afternoon. So MS-STATS provides these tools to both visualize and actually perform this normalization. Okay, so this is one part. And then the kind of core of what MS-STATS does is statistical modeling. And the important thing here is that we try to model all the data for a particular protein which was collected as part of the experiment, meaning all the time points in the time course or all the samples in a paired design experiment, label-free, label-based, right? So reference and endogenous. So everything which pertains to this protein, we will consider jointly and use all this data to derive some conclusions about this protein. So, and for this, we consider experimental design, we consider biological variation, and also the technological variation, okay? So here's just a little bit of intuition. So what we're really doing by extending the t-test to a more realistic and more common situation. So one extension is working with more complicated designs like the one I just mentioned. So here's another example where we work with samples from 5 disease groups from cardiovascular disease. And so this would be a cartoon which kind of mimics what we had with the t-test, but now we have 5 groups and not 2 groups. So in principle, we can consider each pair of groups separately, and do exactly the same thing as we discussed before, right? Consider the means, consider the variability between the means, and so on. But since we have 5 groups, we can do better. Because even if we are interested maybe in these 2 groups, we can make assumptions that around the mean, the corresponding variability has something in common. A very simple assumption is that the variance around the mean is exactly the same across all the groups. Or maybe it has the same relationship to the intensity, or maybe it has some. So as long as there are some kind of shared patterns which are consistent and repeatable across the groups, we can learn these patterns, and then we can learn the variability around these means and inject this in the denominator of the test statistic when comparing these 2 groups. So for example, if we can assume that all these variances are the same, then we can combine the estimate of variation across all the groups. And when we compare these 2 groups, we have better estimation of variability because we borrowed the information on the variability from the other groups. And this is so we mentioned ANOVA last time. This is exactly what ANOVA will do if you have very kind of perfect situation, no missing values and so on. More generally, linear mixed models learn these patterns of variation, and they inject the information on the variation from this group into a comparison of any pair of conditions. So in a situation where you have multiple related conditions and they're measured jointly, so it kind of works like a kind of extra sample size. So even though they come from different conditions, if you can borrow the information variation, it kind of as if you had more replicates in the conditions of interest. So it's actually really advantageous to model multiple conditions together. And it's a good strategy, provided that these assumptions are actually reasonable. So this is one thing, and so this is how you would do it. So you would just explain how the variability in the data is explained by the deviations from each group from the overall mean, and then you have some random deviation here. And so this is another kind of representation of also of the same aspect of experimental design. So if you have 2 groups, so healthy and disease, so this is the range of values that you expect in the population. You measure biological replicates from these populations, and then the black dots are the technical replicates from the biological replicates. So you can also kind of represent with these models what comes from the biological variation and what comes from the technical variation. So again, you can extend the models which were used for the t-test to show that there are multiple layers of variation. Okay. So that's very useful in that sense. So this is experimental design, right? So this shows how we can account for multiple sources of biological variation and leverage them to make better conclusions. But there's also the technical, the aspects of technical variation. And so this is what a label-free SRM experiment would look like. So let's say we have condition 1, condition 2. Within each condition, we have multiple subjects. Maybe within each subject, we have multiple runs. So this could be technical replicates. But then we also have multiple SRM features or peptides, right, which pertain to this protein, to this subject within the run. And so, well, now different features, they have different ionization efficiency, they have different fragmentation patterns, they have different kind of interference types, right? So now we also need to integrate the information not only across the condition subject that runs, but also across multiple features. And in fact, what you can do, you can extend linear models further to kind of say, okay, this is the part which reflects the biological variation that comes from the experimental design, and this is the part which reflects the information from different features. And so if you have been working with MSTATs in the past, so MSTATs version 2 kind of looked at all of this together. So MSTATs version 3 now kind of developed an approach where you can partition the biological variation and the technical variation, account for technical variation first, and then kind of use this to further test hypotheses or quantify proteins on the biological variation. And that is really useful because it allows us to be even more general than before. It also allows us to be faster than before. So this is kind of a big change in how we view this data now in the new version of MSTATs. And so the same thing for labeled experiments now, right? So if we have labeled reference peptides, so we have even more data because now we have the endogenous features and we also have the reference features which correspond to them. And so again, we can further extend these models to account also for all of the sources of variation and then combine this information together. So it becomes eventually fairly technical, but the message is that it's just an extension of the t-test which pulls together more different sources of information in the right way. And the good news is that it's all done in the background, meaning that you don't have to specify these models. All you need to say is, I'm interested in comparing this group to this group, or maybe this group to the average of these groups, and everything else is done in the background. Okay, and so after we do this, well, we produce this type of volcano plots that we just discussed, right, with the practical significance and statistical significance. And so adjusted for false discovery rate, so exactly the same way as what we did in the simple case. And so the very last part is, well, going back to the question that we had earlier, okay, we've done all of that, so what have we learned? So clearly we learned which proteins are changing, right? We can use this for additional kind of downstream analysis. But we can also use this data to learn about the extent of variability that we have in our system. And we can use this to design better experiments. Next time we have to repeat a similar type of experiment, maybe as a confirmatory study or as maybe some other related question and so on. And typically MS Stats uses every data set which is being analyzed. Immediately it uses it as if it was a pilot study for some other future experiment. And so what we can do when we kind of work with this data, so we estimate all the variance components, we can use these variance components to start kind of predicting what would be the trade-off between fold changes, sample sizes, and different extent of variation. And we would make plots like that, where this will be the desired fold change that we want to see in the future experiment, and this is the number of biological replicates that we would need for that. And so one aspect which is often overlooked is that this relationship between the fold change and number of replicates also depends on how many proteins you are actually expecting to see changing. For example, if you are working with a DIA experiment and you expect that only a small percentage of proteins will be changing, typically you would actually need more replicates than if you do a targeted experiment where you expect many true changes. And the reason is, again, multiple testing that we talked about before. Because remember, we control for false discovery rate, meaning that we control the probability of making false positive discoveries. Now, if we are in the experiment where everything is a true change, we have less opportunity to make mistake. So we don't need to work that hard. On the other hand, if we're in the experiment where we have many proteins and only few of them are changing, we have a lot of opportunity to make mistake. So then we need to work harder to protect ourselves against this mistake. And work harder in this case means you need more biological replicates. And so this is kind of an illustration of that. This will be the sample size you would need if you only had one protein. This is the sample size you would need if you had proteins and 50% of them would be true changes. And this is the sample size you would need if you have proteins where only 1% will be true changes. So you will have more opportunity to make false positives, so you would need to protect yourselves with that, from that with the larger sample size. And so to calculate that, you actually need quite a few things. So this would be what MS Stats would require for that. So you would say, okay, here is the false discovery rate that I would like to control, right, the level of FDR. This is the proportion, the ratio, or you can set it up as a proportion of features which you expect to be true changes. And clearly for targeted experiments, it will be very different from kind of global or DIA experiments. Statistical power, so the probability of actually seeing the change when it's a true change, and it's typically set to something like 80%, so that's kind of standard. Anticipated fold change, well, 1.1, 1.2, 2, and so on. So all of these things you can kind of have just kind of from common sense, from literature, from kind of general kind of way of doing things. This is the hard part. You actually need to have the variance components, so the amount of biological variation, the amount of technical variation that you expect to see in the future experiment. And this is very experiment, workflow, system specific. It's very hard to make up these numbers kind of from nothing. And so usually this is why when we analyze a data set, we immediately kind of get these variance components to use them as the input for planning a future experiment. And this is why when somebody comes to me and says, look, I have no idea what I'm doing, but can I use 3 patients? I say, I don't know, because it depends on that. So if we don't know how much noise, and by noise I mean both biological variation and technical variation we have in the system, it is really very hard to say if 3 replicates will be enough or not. And that goes back to Edward's question, right? Are the experiments underpowered or not? Well, it depends on the interplay between fold change, variability, how many things are changing altogether. So it's kind of complex thing. And in fact, to design a good experiment, you actually need to know a lot about your system. And this is why pilot experiments are generally a very good idea, because you can learn a lot from that, not just from the workflow viewpoint, but also kind of statistical aspects, quite a bit. Okay, so that's the end of that. So I just wanted to advertise once again. So this is a very good colleague of mine, Naomi Altman, who is doing really good work trying to explain fundamental concepts in statistics in this Nature Method series. So I would very much encourage you to look into that. And I think we have maybe a minute or two for questions.
Yeah, exactly. Thanks, Olga. Other questions?
No questions? I hope I didn't scare you. All right. Well, I'm around here. Oh, okay.
My question, I guess, is only loosely related. It has to do with missing data. And how do you, basically, what's the ideal approach from a statistical standpoint to deal with those?
Very good question. Actually, yeah, I was hoping I would talk about that. So, well, you know, kind of generally speaking, right? So if you don't have the data, missing data means you have no data, right? And so the only substitute to data are the assumptions, right? So the only thing you could do when you don't have the data is to inject some kind of assumption about why you don't have it, right? And so you can do it in multiple ways. You can assume that it's not there because it's missing at random. You can also assume it's not there because it was below the limit of detection. It depends how you do the signal processing. For example, Skyline actually distinguishes these two cases, right? And it gives you 0 or NA. NA means that it's missing at random. 0, it means that it was below the detection level. Other software tools would not do that, so you kind of have to figure it out yourself or just treat everything as you don't know, right? So once you have that, you have options, right? So, for example, if it's missing at random and if you really think there is no kind of reason it was just failed for some reason, right? So it is actually reasonable to leave it missing, because linear models can accommodate this type of things in a kind of good way. Because so linear models can handle this unbalanced design where you have holes in this kind of data structure. Okay. Now, if you have censored values, meaning that they were below the detection limit, you can do several things. You can do imputation. So you can assume that you just have the lowest value in the run, or the lowest value in the feature, or integrate the noise in the area where you expect to see the peak, and use that. So, and this is one option, by the way, that we offer in MStats. Now that we're kind of in the version MStats 3.0, now that we split this kind of by treating separately the biological and the technical aspects of the data, it allows us to be more creative with how we handle missing values. And so a new option that we offer is actually explicitly modeling censoring. So censoring means that I know that the missing value is below some threshold. I don't know what it is, but I know it's not above, I know that it's below. And when you estimate the parameters of this linear model, you can actually account for that. It's not easy because you still need to know what's your censoring threshold, right? And that may be very specific to different peptides, right? So it's still not 100%, but there is evidence that this is also a very good way to do this. And so the new version of MStats has that. So you have option to treat this as censored, and especially when signals are processed with Skyline, we can kind of rely on that when Skyline kind of annotates this as censored. You can impute, you can leave it as missing. And for the moment, a good strategy, I would say, is to use all of the above and see how your results depend on that. And so if you feel that your results are very much depending on how you handle missing values, you should probably go back to the raw data and see which of those situations is the most plausible. If, on the other hand, whatever you do doesn't matter, well, then that's your conclusion.
Can you say something about how to parameterize the imputation? Or how, I mean,
Say this again?
The imputation, where do these values come from? This is a simulation, and how do...
Oh, there are many ways of doing this, and there are complicated ways and simple ways. So a simple way would be to just have, as I just said, the lowest value for this feature across runs, the lowest value for the run across all the features, or if you can have access to the signal processing tool, integrate the noise in the area where, and just use the noise kind of integration as the value, right? So this will be simple ways. And frankly, that's what I would go with. I wouldn't do anything more complicated. But some other people would do things like they will look for other features which have exactly similar profiles and use the pattern from these features to impute. But then you can use your data many times over, and I'm not sure it's really worthwhile.
But the lowest value imputation, then you have the same number in a lot of fields, right? Doesn't this cause problem in the group comparison then?
Well,
with
but
respect
that's
to the
then
variances?
you are in this situation, right? If I can go back here, right? So you just start with something which looks very irregular, but if the sample size goes up, you can still work with that. Yeah.
Yeah, I guess that's for your talking. That's a major concern about the diagnostic biomarker, which is they only look at the protein levels changes between two conditions, which is disease and
or more conditions, right? So as I was saying, right, you can actually do any combination, any linear combination of conditions, right?
Yeah, my question is that if you're looking at something like predictive biomarkers for a drug, for example, if I want to look at, say, the 20 or 30 biomarker or protein changes against the drug efficacy, and to establish whether these multivariate analysis is anything that MS stats can do, or you can suggest any other software can do.
Yes and no. So I wish I had another hour. I could tell you all about that. So yes and no. So what you want to do still is to summarize the information that you have from this type of kind of complicated structures, right? Multiple features, multiple conditions, multiple runs. And so MS stats can do that. So I kind of emphasize the fact that you summarize differences between the groups, but you can also say, I want to summarize all the numbers which pertain to this particular subject across maybe all the technical replicates, all the features, and so on. So this is what is called subject quantification in MS stats. And you kind of use a similar procedure. So MS stats will give you a summary which accounts for missing values or some kind of holes in the data, which gives you one number per protein per subject. Okay? So this will be summarizing multiple features, label-based, label-free, right? So it will be all kind of taken together. But of course, this is not enough, because now what you need to do is to develop a predictive model which predicts the status of the disease for the individual patient. And for that, you would need to do more work, and so it will be a supervised method. So this is class prediction, right? So my third statistical objective that we talked about yesterday, methods like logistic regression, linear discriminant analysis, support vector machine, random forest, right? Whatever you like to do. And you would use this number per protein per subject as a feature which can potentially be predictive. And then you have to be very careful in terms of selecting the features, making sure you have a training set, you have the validation set, you evaluate the error rates, such as false positive sensitivity, specificity, positive predictive value. So you have to do all this work. So MS stats can help you by summarizing all of the features and labels and replicate runs into one number per protein per subject that you use as input to these machine learning tools. And then, well, R has plenty of those, but not only R, there are many other tools for that. So MS stats does not implement any of the predictive models. We'll see, maybe in the future, no, not now. Yeah.
So no other questions. Thanks, Olga, again.
So I will be around, so if you have more questions later, so let me know. Okay. Thank you.